Thank you, Shuka, and welcome everyone. Glad you all could make it here. So just to start us off, we've got a session on modified channels. We've got 3 talks lined up. This is a topic that's near and dear to my heart, as well as I think many of you. Our first speaker is Dr. AJ Reisinger from the University of Florida. I heard him give a great talk on the subject of artificial ecosystems at the Society for Freshwater Science meeting in Brisbane, and I thought it'd be actually really great for sharing with the audience in California. AJ, are you, I saw you're logged in. Are you able to unmute yourself and share with us?
Slide. So I'd like to thank the members of my lab that have really helped do a lot of the sampling that I'm going to be talking about here today, particularly 3 undergraduate students, in addition to the co-authors listed on the talk. Sam Howley, Annabelle Schreiber, and Katie Schoenberger have all contributed a lot to this. Collaborators at the University of Florida and beyond that have helped us advance our understanding of artificial aquatic ecosystems, and then funding sources. Much of what I'm going to be talking about today is either currently or previously funded by the National Science Foundation, the Florida Department of Environmental Protection, and then a few internal University of Florida sponsors. Next slide, please. Before we talk about some specific work that we've been doing in our lab looking at artificial aquatic ecosystems, I also want to just kind of introduce the topic of artificial aquatic ecosystems to those of you that might not think about it as much as Raphael and I. If you're not familiar or haven't studied artificial aquatic ecosystems, I highly recommend checking out this review article by Chelsea Clifford and Jim Heffernan that was published in 2018, where they do a really great overview of all of the different types of artificial aquatic ecosystems that are prevalent on our landscapes. They span from accidental to deliberately created ecosystems, and those that are simply altered or completely constructed. So this can include anything ranging from artificial wetlands and accidental wetlands in Phoenix that were studied really heavily by Monica Paulton. Was kind of my first aha moment, was thinking about these accidental wetlands, or roadside ditches and gutters and curbs that were studied by Annika Bratt in the greater Minneapolis-St. Paul region, and all of the decomposition that's occurring in these curbs and gutter systems. There's been work looking at the downspouts off of roofs that drain gutters that have this really unique dissolved organic matter composition from the allochthonous leaf litter inputs into the gutters, or 100% effluent driven streams in southwestern US that are completely dry if it were not for wastewater treatment plant effluent discharging into these streams. Finally, there's heavily engineered ecosystems like the Los Angeles River, which I'm sure many of you are more familiar with than I am. Or a common thing in the southeastern United States in agricultural areas are tailwater recovery ponds. And these are human-made ponds that are dug out to capture excess irrigation water that's not used by the crops, and is then returned back into a canal system that allows for future irrigation, kind of reuse that irrigation water. Finally, we've manipulated a lot of these aquatic ecosystems to make them more or less green, depending on what we want from them. This can include things like aesthetic manipulations that we often see in a lot of parts of the country, particularly in Florida, for like golf course ponds. Or there's more functional ecological manipulations through things like floating wetlands or other constructed treatment wetlands within some artificial ecosystems, like is shown in the bottom left. Regardless, this doesn't give you the entire every single artificial aquatic ecosystem, but it gives you a nice little overview of the range of what these ecosystems might look like. And whenever I am talking about this with some of my ecology colleagues, they say, oh, those aren't real ecosystems. Why do we need to study them? And so I think that it's really important to push back against this narrative that if you're working in these concrete ditches or these ag streams, that the ecology isn't as important. These anthropized ecosystems is a term I really like to use. Anthropized ecosystems are those that are designed, constructed, and managed to provide specific benefits to society. Sometimes they're termed engineered ecosystems, sometimes artificial, sometimes designer, but regardless, they're designed for specific purposes. For example, let me get my laser pointer here. Reservoirs are designed for maybe energy generation or drinking water collection and storage. Agricultural areas are designed for food production. Bioretention cells in urban landscapes are often designed to capture excess surface water runoff. And then irrigation canals are designed to move water back and forth and drain things that we don't want to be wet and provide water to things that we want to be wetter. However, they're also expected or presumed to provide secondary services as well. For example, reservoirs are often expected to provide some recreational services like boating for society. Agricultural areas are recently expected to provide carbon sequestration in the soil. Bioretention cells are expected to provide pollinator biodiversity in urban landscapes. And irrigation canals are expected to potentially provide, I've seen even solar panels over top of them. They do provide irrigation sources as well as drainage. Because of their designed, engineered aspects of these systems, they're often overlooked by ecologists. And like I said, I have gotten the specific comment in papers that I've published that these aren't real ecosystems, so why are you trying to apply real ecological principles? And I want to, again, focus on the real ecology that's happening in these artificial ecosystems because they're everywhere on the landscape. This is a Google Maps overhead view of the city of Gainesville, Florida, which is where I live. My house is out in this area of town, kind of on the suburbs. You can see here, this is the swamp, our college football field. I'm presently located about right here. So this is an overhead view. You can see the interstate running by a few major roads. Now, if I overlay the stormwater pond system, the stormwater ponds in the city and the greater county area, there's about 800 stormwater ponds throughout the greater Gainesville area. These include both dry and wet ponds, but we get a lot of rain here in Florida. So these ponds have a lot of aquatic ecology going on. In addition to these stormwater ponds, we have to figure out a way. We built these ponds to capture excess runoff from our landscape. So therefore, we have to actually get that excess runoff to them. And we do that through our really complicated network of surface and subsurface channels. So shown on here, this is the city's stormwater conveyance network. In green are any surface channels or surface drainages, and in blue are the subsurface pipes. So you can see there's a lot of artificial channels, artificial flow paths, but just because we're artificially engineering these systems doesn't mean that water and the things that are responding to water don't occur in these environments. I like to point this out because we as a society, we're really good plumbers. We're really good at moving water from where we don't want it to where we want it. And that can have a lot of significant influences on watershed processes as well as within a specific ecosystem, a specific reach. So I know what you might be thinking. I'm an ecologist, not a plumber. And so why should we care about where this water is going? Again, I'm hopefully trying to convince you that this is a really important thing. But beyond just the ecological processes, it's important to understand how this plumbing of our watersheds is affecting the environment, because artificial water bodies are the most commonly experienced aquatic ecosystem by society. At least that's the case for Florida. So the figure at the top right here are results of a survey of residents of Florida, and they were asked how frequently they encountered a range of different aquatic ecosystems. And this is ordered from the most frequently encountered to the least frequently encountered aquatic ecosystem, with the black being encountered less than once per month, and the light gray being twice weekly or more. So stormwater ponds are by far the most frequently encountered aquatic ecosystem by residents of Florida. Lakes and rivers are also frequently encountered, but so are canals, and they're encountered more frequently than oceans, streams, or our really idyllic springs that we have. And it's for this frequency of encounter is likely because we're getting rid of a lot of our natural aquatic ecosystems on the landscape. We're losing our natural streams and channels, and that's shown really elegantly on a few figures here on the right from previous work. This is an overhead view or a schematic view of the city of Detroit. With this black outline is the city outline. And you can see in red are all of the buried channels, buried streams in Detroit. And in blue are any surface streams that remain. And so you can see that within the city limits of Detroit, there's only one surface channel that hasn't been buried. And this is common throughout metropolitan areas across the United States and likely beyond, as was shown by Meredith Steele and colleagues in their homogenization of surface water distributions across the United States. They found that with increasing urbanization, you get a reduction in surface channel density, and that was across 9 different major metropolitan regions throughout the United States. So this is showing that this kind of artificiality of our drainage systems and our artificial development of aquatic ecosystems is influencing our natural environments as well. And I think it's really important to get a better understanding of the artificial ecosystem functioning because they are everywhere and they represent this opportunity to regulate biogeochemical cycling and incorporate ecological processes and dynamics into the design, construction, and management of artificial aquatic ecosystems. So we get to choose how we build these. We get to choose how we manage these and how we design these. There's some choices that are made for us by nature. If nature wants to fight back against us, sometimes we can't win. But overall, it's up to us to design these systems. So for the rest of the talk, I'm going to talk about how effective these artificial ecosystems are at processing and removing nitrogen specifically. I'm a biogeochemist really interested in nitrogen and energy cycling. And so the vast majority of the remainder of my talk is going to be on nitrogen cycling. We'll start with stormwater ponds. And stormwater ponds are really prevalent in the state of Florida. We published a paper a few years ago that estimates that there's at least 76,000 stormwater ponds throughout the state, accounting for upwards of 3% of the state's surface area. So there's more stormwater pond coverage in the state than industrial land use. And so they're everywhere, but we don't really know how well they're functioning. In state regulatory language, there's actual state mandates on record in Tallahassee that say that stormwater ponds, if they're built to a certain engineering threshold, they're expected to remove 80% of nitrogen, phosphorus, and sediments that enter into the system. So an 80% removal efficiency of inflow minus or outflow relative to inflow, sorry. And so we know from previous work that these ponds are really good at removing sediment and phosphorus. So this figure on the right is from the Florida Stormwater Bible, as I like to call it, the Harper and Baker report. So they reviewed a vast number of hydrological and biogeochemical studies of stormwater ponds, and they assessed the removal efficiency of phosphorus first, so the percent phosphorus removal based on inflow and outflow concentrations. And they found that if you hold on to water in a stormwater pond, so the detention time of about 200 days, you get to that 80% removal efficiency that's expected by state legislative language. So it's not every pond that's going to get to that 200-day detention time, but it's pretty common. So these ponds they're living up to what their expectations for phosphorus and for sediments as well. However, we know from that same report that ponds aren't very good at removing nitrogen. So the same study, the same review, inflow and outflow concentrations, removal efficiencies of nitrogen. And you can see that even if you hold on to that water for 500, 600 days, you don't really cross the 50% threshold for removal efficiency. You never even come close to that 80% expectation. So ponds, our empirical data show that ponds are not as good at removing nitrogen as we would expect. Why is that? So most of what we've known prior to my coming here and prior to some of other recent work by other colleagues throughout the state on stormwater pond functioning has been based on inflow-outflow concentrations. And there's been kind of black boxing approach of the actual ecological processes occurring within stormwater ponds. And so we really try to open up that black box and look at some of the mechanisms occurring within the ponds. And we found that ponds aren't very good at removing nitrogen because the removal processes are limited by various factors. So doing a study of sediment fluxes of nitrogen, a former master's student of mine, Steve Homan, who now works with the EPA based out of Philadelphia, he was looking at net nitrogen flux, which is the balance of denitrification as a positive nitrogen gas flux and nitrogen fixation as a negative gas flux. So a positive flux means the pond is removing nitrogen from the system, or the sediments are removing nitrogen from the system. Under ambient conditions, he studied 8 ponds in 4 in Gainesville and 4 in Miami, across 2 seasons. So in Florida, we have 2 seasons, not 4. We have the dry and the wet season. And so he found that under ambient conditions, when you expose sediments from these 8 different ponds, there wasn't to ambient site water, there wasn't a lot of net nitrogen gas flux. So there was a little bit of an increase during the wet season with pond age and a little bit of a decrease during the dry season, but really pretty flat lines and not a lot of difference from zero. However, we wanted to see, okay, how would ponds, how the sediments respond to a simulated stormwater runoff event. And so for that, we elevated the inflowing water flowing over these sediment cores by spiking them with 1.2 milligrams of nitrate per liter, which is in line with what we actually measure with stormwater runoff in the region. And we found that when we elevated this nitrate condition, all but one of the sampling events, all but one of the experiments exhibited an increase in denitrification. And this increase in denitrification was particularly prevalent with increasing age in Gainesville. So older ponds got better and better at denitrifying in response to this elevated nitrogen runoff event. We didn't see the same response in our Miami ponds, and so we wanted to know why. Well, when you look at the Miami ponds versus the Gainesville ponds, there's some notable differences in the management. The Gainesville ponds that we studied had a lot more natural vegetation, had a lot more organic matter buildup, had littoral and submerged aquatic vegetation, and kind of wooded area around the edges, around the banks. Contrast that to our Miami ponds that were mowed up to the edge of the ponds. The sediments were basically clear white sand, suggesting that they'd been frequently dredged. And so, although we have a similar age gradient between Gainesville and Miami, we don't have as a similar ecological age grading, if that makes sense, because we think that this management is offsetting this ecological development. But that was just looking at the sediments directly themselves. We wanted to know what's happening in these stormwater ponds at the whole pond level. And so to do that, another grad student in my lab, Audrey Geckner, went out and sampled 21 ponds and natural lakes and ponds in the region. And she studied these in 2 regions. So she sampled 15 stormwater ponds from Bradenton, Florida, which is a suburb of Tampa. And then we found 6 undisturbed natural ponds and small lakes at the Ordway-Swisher Biological Station, which is about an hour east of Gainesville. We had to sample these diverse regions because there aren't really any mini natural ponds or lakes in close proximity to these suburban communities. At all of these ponds, she collected dissolved gas samples from 3 different depths: the surface, middle, and bottom of the lakes or ponds. And from these samples, she quantified dissolved nitrogen gas concentration using membrane inlet mass spectrometry. From those concentrations, she calculated the N2 saturation ratio. So she looked at the measured concentration and compared that to the equilibrium concentration, or what we would expect from dissolved nitrogen gas if there wasn't any biology occurring. So you can figure out what the equilibrium concentration is based on just pressure and temperature. And then if the dissolved nitrogen gas concentration was higher than the equilibrium, that suggests that the system is supersaturated for dissolved nitrogen gas. The way that a system becomes supersaturated is something in the pond is producing nitrogen gas in the pond, and that's through the process of denitrification. If the N2 concentration measured is less than the equilibrium concentration, that tells us that the system is undersaturated or subsaturated with nitrogen gas. So that means that something in the pond is sucking that nitrogen gas out of the water and using it for something else. And that's through the process of net nitrogen fixation. So anything above 1 for these ratios is suggesting that the pond is a net benefit for water quality and removing excess nitrogen, whereas anything less than 1 suggests that the pond is actually producing nitrogen in excess of what's coming into the system. Okay, so just jumping into the results here, we're looking at the N2 saturation ratio on the x-axis and then just the frequency of occurrence on the y. The natural ponds are shown in green and the urban systems are shown in the light gray. So you can see that there's kind of a pretty decent balance of both nitrogen fixation and denitrification across all systems. But if you actually dig into it, we found that natural ponds and lakes, 80% of those samples indicated net denitrification and 20% indicated net nitrogen fixation. So on the whole, natural ponds and lakes were predominantly removing nitrogen from the system and a benefit to water quality downstream by removing nitrogen. Urban stormwater ponds, on the other hand, had nearly perfectly 50-50 split between net nitrogen fixation and net denitrification. So some urban stormwater ponds were removing nitrogen and protecting downstream water quality, but an equal amount were actually producing nitrogen within the system and exporting more inorganic nitrogen than they received from the inflows, indicating that they're a net source of nitrogen for downstream. So that tells us that stormwater ponds are not mimicking the natural biogeochemical functioning of the natural lakes and streams in the region. We wanted to know why that is. Well, that doesn't seem to make sense at first, but when we actually dug into it, we looked at the data and we were like, oh, that's interesting. We found that under ambient conditions, nitrate in our stormwater ponds is actually below detection more often than not. So our detection limits in our lab are 6 micrograms of nitrate N per liter. So that's a pretty low detection limit. And frequently we aren't able to detect nitrate in these systems. And so we think that that combined with the fact that our nitrate pulses increased our denitrification rates, but it also increased nitrogen, inorganic nitrogen flux into the sediments. That tells us that nitrogen is actually limiting these processes and that assimilatory uptake is outcompeting denitrification in these systems. So it doesn't quite make sense for urban, heavily culturally eutrophied system to actually be nitrogen limited, but we think that's what's happening, and that's why we are actually seeing this increase or not as good of a removal at the ecosystem scale for nitrogen removal efficiency, just because they're nitrogen limited relative to like phosphorus availability and that sort of thing. We also see in the water column, we see nitrogen fixation across the board in the water column. We did a nutrient addition bioassay and measured net nitrogen processing rates across control, phosphorus, and then various nitrogen plus phosphorus treatments combined. And we found pretty much across the board, we get consistent nitrogen fixation in the water column. So that's telling us that there are phytoplankton, cyanobacteria, something in there that is fixing nitrogen. And that's also contributing to the lack of total nitrogen removal efficiency as shown by the prior work at the inflow-outflow concentration perspective. All right. In addition, we know that stormwater ponds aren't only nitrogen and phosphorus stories. We also have found that they're really good at burying carbon. Organic carbon stock increases with pond age. So looking across a 14 to 35 year stormwater pond age gradient, we see a pretty linear increase in organic carbon concentrations in our sediments. But we also find that the burial rate, so not only is the stock or the concentration increase, the rate that's changing over time is increasing as well. So not only are they sequestering or storing carbon in the sediments, but they're getting more and more efficient with it as these ponds age. However, they're really good at emitting carbon too. So particularly younger ponds, earlier in the development or successional sequence of ponds, they emit a lot of carbon, and that leads to these ponds being net carbon sources to the atmosphere via their high emissions. So the red dots here are the net emissions or sinks of carbon and CO2 equivalents, and on the green are our emissions, and light red is our burial and sediments. You can see that as ponds get to about 25 or 30 years old, they do start to become net sinks of carbon, but it takes a long time to get to that point. And that tells us a lot about how our management might be affecting these systems. So I told you earlier that the ponds in Miami looked like they'd been dredged pretty repeatedly. And this is just another visualization of what that dredging might do. This is a sediment core from a 3-year-old pond. You can see the sand that was installed like during the construction of the ponds, and then about 3 years worth of organic carbon accumulation in the system. And compare that to a 23-year-old pond, you can still see that sand layer that was there present during construction. And then you can see how much 23 years of carbon accumulation looks like. So now just think about what would happen if you dredged all of that muck out, what that does for carbon accumulation, carbon sequestration, but also carbon that's fueling some of our other biogeochemical processes that we're interested in. Okay, so like just to kind of summarize that, we do see that ponds can be biogeochemical regulators. They can sequester carbon. They can actually remove nitrogen from the systems, but they're not going to do it on their own. They're not going to do it passively. We need to manage them correctly. All right, so this is some new stuff that I added in after Raphael asked me to make a little bit more of a plug for some flowing systems as well. And so I want to talk to you a little bit about the South Florida watershed. The South Florida watershed is, I mean, it's the Florida man of any ecological system or any watershed. South Florida is a whole world in and of itself. It's got major cities, including Palm Beach, Fort Lauderdale, and Miami. It's got a lot of agriculture, tropical fruits like mangoes, vegetables, and sugarcane. And then there's really beautiful and really interesting natural areas like Lake Okeechobee, Everglades National Park, and Biscayne National Park. So there's a lot of conflicts and competing interests down there, ecologically, socially, economically, etc. So that leads to a lot of plumbing to move the water where we want. Like I mentioned earlier, we're really good plumbers, and we've probably done more as a society to plumb our watershed in South Florida than maybe anywhere else in the United States, maybe anywhere else in the world. I don't know. It's definitely up there on the top 5, at least, I'd say. Historically, slow surface water would flow through creeks, sloughs, and marshes, flow through the Everglades National Park, and eventually make its way to coastal environments. Natural habitats along those flow paths removed nutrients, settled as suspended material, promoted groundwater recharge, and controlled flooding. However, the canal network was built in order to help distribute the water at the quality, quantity, timing, and location that we desired it. So we wanted to drain places that we wanted to farm. We wanted to move water for irrigation that we needed to use. We wanted to move water away from places that we wanted to develop for our houses. We replumbed the watershed for our purposes. And that has a lot of implications for where that water is going and what is happening to that water along the way. My colleague, Ashley Smith, who's a co-author on this talk, she's a coastal biogeochemist at the University of Florida, but she's actually based in Homestead, which is kind of right down here. It's south of Miami. This is a schematic view of the Biscayne Bay region. Biscayne Bay is broken into a North Bay, a Central Bay, and a South Bay, with Miami being kind of the North Bay, and these are the barrier islands that are all highly developed. But there's also a lot of agricultural inputs coming in from what's known as the Everglades agricultural area, and canals draining that system, making their way into the South Bay and Central Bay portions of Biscayne. We have seen over the past couple of decades that concentrations have been increasing for both chlorophyll A as well as nutrients. You really see spikes in chlorophyll A concentrations in the North Bay and the urban areas, but you see nutrient increases, high levels in the agricultural parts of the South and Central Bays. So this is telling us that the nutrient inputs are coming in one place and they're being used in another place. Maybe there's some bay-wide circulation. I'm not a coastal hydrologist, so I don't really know the circulation pathways here. But there's a lot of concern for the nutrient inputs and what that's doing to chlorophyll A, as well as subsequent responses with things like fish kills, hypoxia, harmful algal blooms, that sort of thing. We know that agricultural land use is a strong predictor of nitrate loads. If we do basin-wide mass balance estimates, we find that urban basins contribute about 35,000 to 80,000 kilograms of nitrate per year to Biscayne Bay, whereas agricultural basins contribute about double that, 80,000 to 160,000 kilograms of nitrogen per year. And a lot of those contributions are coming through our canals. So water is moving from the agricultural areas, from the urban areas to the bays via these canals. And so Ashley and her group have set up a monthly water quality sampling locations at 9 canal spots along 2 major canal kind of networks that flow through a Redland agricultural area, through the city of Homestead, and into Biscayne Bay. So looking here at the map, you can see that the light green is more naturalized area, natural areas. The orangish is agricultural, and then we see the buildup here as well. And so I'm not going to get as in depth into this study because it's still kind of in the infancy of figuring out what's actually going on. But if we just look at nitrate concentrations from the period of record that they have, they've broken their canals into 4 different land uses. They have the Everglades National Park. So these are canals that are draining natural areas, they have some agricultural influences in the watershed, but they're in the Everglades national protection area. Then there's canals in agricultural areas, canals in urban areas downstream, and then South Everglades National Protection Area, National Preserve, sorry, I can't remember the acronym there, is after this agricultural and urban influence. So this is showing that you get this human inputs, human increases in nitrate, but if you allow the canals to flow through natural areas again, they can process and remove a lot of these chemicals. However, all of the sampling is done under base flow conditions, so it's unclear what might be happening during storm flow events as well. So, like I said, that was a really short kind of summary of what canals can do, but we do know that canals can affect nitrate concentrations, but only if we view them as more than just pipes. So I just want to kind of end with a plug to what we're hoping to do moving forward, particularly with stormwater ponds. We've done a lot of work trying to open up the black box of the biogeochemistry within these ponds, but our next step is to further investigate the mechanisms, the linkages between the social expectations of what these ponds should do for us and how that affects our biogeochemical processing. So we have an NSF DICES funded project that's looking at societal expectations of stormwater ponds at residential, community, and regulatory levels, how those expectations change the management of the ponds. Do higher residential expectations lead to more algal control methods, for example? And then what do those management actions do for biogeochemical and microbial responses? Ultimately, we're hoping to be able to put some economic valuation onto how much stormwater pond water quality is worth and link that worth back to the functioning and management perspectives. So to summarize, because we got started a little bit late and I'm on a little bit of a time crunch as well, I'm going to leave the summary up here. I'd be happy to hang out for a few minutes for questions, but if we need to move on due to the time frame, I'm happy to also answer any questions via email or via social media. Again, thanks a lot for inviting me and thanks a lot for your interest. I'd love to hear more from people in California that are thinking about similar artificial aquatic ecosystems and how their management is affecting their function. So that's all I've got. Thanks a lot.
Thank you so much. Do you folks have any questions for AJ? Hi.
Hi, AJ. My name is Kylie. I'm a tech here at Squirp, and I just had a question. If you looked into ammonia or nitrite levels, or were you just simply looking at nitrogen gas and nitrate?
Yeah, that's a good question. Whenever we collect just background water chemistry samples as predictors, we definitely run ammonium as well. We run nitrite and nitrate, so we run NOx as our method, so we don't disentangle nitrite and nitrate directly. Ammonium levels are higher in stormwater ponds than I'm used to in streams because you have the different redox chemistries going on. So there is some interconversion between ammonium and nitrate and linked coupled nitrification, denitrification going on likely. And also total nitrogen levels are actually quite high in a lot of these systems because you've got a lot of organic nitrogen as well. So yeah, I guess we have that information. It's unclear how that contributes to like assimilatory versus dissimilatory processes, though. That's something that would be worth looking into. Yeah.
Yes, hello. My name is Gaurav Rajan. I'm with the Region 8 Santana Regional Water Quality Control Boards. I'd like to congratulate you. Very interesting paper, great insights. And so I'm really looking for your comments on this. You know, it seems to me the insights you provided about natural, and then the engineered systems, might make a case for more diversity in these urban stormwater ponds. If you had a greater diversity of plants, greater diversity of microorganisms, you may have more of the plants using the nitrogen, versus the ones fixing the nitrogen. Same with carbon. Some would use the methane, some would convert it to carbon dioxide, some would convert carbon dioxide to methane. So that might also tie into the age issue and the dredging. So I'd love to hear comments on that. Thank you.
Yeah, absolutely. Diversity of plants is something we think a lot about. We have a project that I didn't talk about today, but looking at how plantings of stormwater ponds are influencing water quality and also how those plantings are accepted or not by residents. And what we find is that seems to be a sweet spot of letting something grow wild but not too wild that can hopefully couple or like kind of balance ecological functionality with people actually accepting it and not demanding it all be mowed down and cut away. So I think that there is this trade-off of diversity and function, but also we can't let things go too wild when we're working with homeowners, because otherwise they're just going to demand that we clear cut it all. And then you end up getting people fertilizing turf grass up to the edge of their stormwater pond. And that's a whole other story. In our upcoming work with the NSF funding, we are looking at microbial diversity to look at if there's like algal community diversity or bacterial archaeal diversity and what that does to some of the functional processes. We've also looked at how plant vegetation and structural complexity influences macroinvertebrate diversity. And again, we find kind of this sweet spot where super complex, super unmanaged systems actually have lower aquatic invertebrate diversity, because you actually end up with like really low dissolved oxygen, really poor conditions for macroinvertebrates to survive. So there's this kind of sweet spot that we need to find and figure out how to educate the public to accept that as well.
So yeah, I'd just like to talk to you a little bit about a project I'm involved with, the Stormwater Monitoring Coalition of Southern California, focusing on ways that we can improve biological conditions in modified channels, like some of those channels that AJ talked about at the end of the talk. So modified channels are, very widespread in California, no less so here than in Florida. There are certain parts of the state where they're pretty much the only type of waterway you're going to find, like most of the Central Valley and the Imperial Valley, but they're also super common in our urban areas like Southern California. This is from Ventura County, part of the Cayugas watershed. And we've been, for the most part, when we've been studying modified channels, we've been trying to look at the diversity of forms, looking primarily at bed and bank material, looking at things like, is the stream bed hardened? Is there hardening on any of the banks, the left bank, right bank, or both? And we found that this has actually a really good relationship with the types of biological functions that you can see supported in that reach. Last year, I presented some work where we looked at the relationship between our biointegrity index scores in these different channel types, looking at our algae index or the algal stream condition index, or ASCII, versus our bug index, the California Stream Condition Index, or CSCI. And we're finding things that the bug index tends to be very limited, very low in our hard bottom channels, whereas when you look at our algal index, it's really the soft bottom channels where there appears to be these really strong limitations. So we've done a lot of work focusing on, how limited are these systems? How bad do they look? And we kind of were frustrated with that because we want to see, like, what else can we go beyond that? Like, what can we do to start improving the scores? And so the SMC has funded a new project that is really trying to identify some, actionable things we can do that will lead to better conditions in these systems. And we sort of have this two-part study. We want to know, what can we do to improve conditions within these existing forms? And also, what can you do about restoring natural forms? What can you do to improve conditions? So we have sort of multiple aims in this project. So I'm going to talk a little bit about, what are we doing, within existing forms? And really what we're trying to do is apply causal assessment principles to our low-scoring modified channels. Causal assessment, it's a process of, evaluating data and generating evidence to see what stressors are associated with poor conditions at a site of interest. The US EPA has developed a causal assessment framework known as CADIS. That's the Causal Assessment Decision and Diagnosis Information System. It's a really great process. We've done some test cases here in California, but we also recognize that it's a fairly intensive process, takes a lot of time. It's something that we couldn't realistically apply to the sheer number of sites that we have in California that are, in poor condition that we want to fix. And so to address that, we've been developing an alternative called the Rapid Screening Causal Assessment, or RSCA. It's based on the same principles as the EPA's CADIS, but we've kind of streamlined and modified, made some like standardizations to really make it easier to apply to a large number of sites very rapidly. It still does the same kind of analysis. We're looking at data from our test sites where we have low scores and comparing them to environmentally similar sites that we call comparator sites. And what this lets us do is we can take advantage of these large regional data sets we've been collecting, either through the perennial stream assessment statewide or our regional programs like the one that the SMC runs or the Bay Area Regional Monitoring Program. And we can apply this to many sites at once. And this, first of all, it helps us, make some rapid prioritization decisions. We can sort of figure out which sites we want to investigate more closely. And when we do investigate those sites, we're sort of not starting at the ground zero. We have more information, so we know how to, approach those more effectively. when we look at RSEA, we were looking at a standardized set of stressors. The ones that we focused on are eutrophication, temperature alteration, physical habitat degradation, and salinization or elevated conductivity. We've standardized the indicators of those stressors. So these aren't all the things that can occur in a modified channel in California, but we do know from a lot of our past research that this covers a large number of the types of stressors we often find in our streams. But there are, of course, other things like invasive species or pesticides or other types of toxicants, as well as hydrologic alteration, which is a topic you'll actually hear about more in our next talk. We also once we have the data we've identified, we have a standardized approach for evaluating it. We generate data by comparing the data at our test site to comparator sites. We might ask questions like, are stress levels lower at healthier sites? And so you can sort of see a little example here with this box plot, if that's showing the range of stress values at your healthier comparator sites. If you have higher stress at your test site, that's supporting evidence that that stressor could be responsible. If it's lower than your healthier sites, you would say that's weakening evidence. It's probably not the problem. If it's in the middle, you say that's indeterminate. You can't be sure one way or the other. We have a couple of different lines of evidence that we look at. Some are based on this type of analysis. Others are based more on like dose response models, but they're basically doing the same thing of trying to figure out whether the levels at your test site make a good case for whether that stressor is the problem. So we have this rapid approach, and we've been able to apply it to a large number of modified channels in Southern California. We have, well over 100 sites that we've figured out, where we've collected bioassessment data, and we figured out, what kind of modifications are going on. We have a whole lot of hard bottom channels, particularly in the LA area, but we do have soft bottom channels that have been engineered throughout the region. And so basically, we've been able to now start to draw conclusions based on doing this sort of large-scale causal assessment. I'm just going to walk you through some examples looking at natural channels first. Now, we find that about half of our natural channels are in good condition. That's that purple block on the right says passing CSCI. So you don't really need to do any causal assessment. There's no problems there. And of the remaining sites, you find evidence supporting one of those stressors at about 25% total. So there isn't one stressor that's really sticking out as being the major problem at these natural channels. Now I'm showing you the results for all the different channel types. And you actually start to see some really interesting patterns. And if you look at the one at the bottom, the panel at the bottom, these are the hard bottom channels. We're finding evidence that nearly every hard bottom channel in Southern California where you have a poor CSCI score, that problem is likely associated with temperature alteration and habitat degradation. Now, the habitat degradation is not really much of a problem. You harden the stream bed, there's probably something going on with the habitat. The temperature also makes sense. You're often getting rid of the shading. You're often making these streams really shallow, so they heat up really fast. So it's not too surprising that we're seeing temperature as a potential problem. When I look at eutrophication and conductivity, these are what I'm calling shorthand as water quality problems. They're affecting about half of the hard bottom channels. Or should I say there's evidence supporting them being an issue at half of those hard bottom channels. And so that's also pretty interesting. It's, they're clearly potentially affecting a lot of these ones, but it's much less common than those other two problems. And when you look at the soft bottom channels, these water quality problems are suddenly a lot more likely relative to what you'd see in the hard bottom channels. And because we're able to look at a lot of sites at once, we can actually start to make some sense out of the spatial patterns, the geographic patterns. And so these maps show you the results for all the different hard bottom channels. The yellow dots are where we're finding likely causes, and the green dots are where we're finding evidence that they're unlikely. And when you look again at those water quality problems, a clear pattern emerges where there's a big cluster of those green dots, particularly in like the LA basin area. So that suggests that, water quality may be less of an issue in that region than in other parts of Southern California. Now you may be asking, you're comparing your site, the data you collect at these modified channels. Some of your comparators a lot of them are probably not modified. They're natural channels. And is that really giving you a useful insight into what's going on? So what we decided to do is, take a look. What would happen if we sort of redid our analyses, but we only tried to compare our modified channels to similarly modified channels? So if you have a hard bottom channel, you want to know how does it compare to higher scoring hard bottom channels. And the idea is that this can help us see if the stressors we're looking at are making a contribution to poor scores beyond simply their association with channel modification. Our thinking is that if a stressor goes from being likely with a traditional assessment to unlikely with this alternative way of assessing, that might tell us that that stressor, it's probably contributing to poor scores, but it's not a limiting factor. And therefore, trying to fix that problem may not have a big payoff. In contrast, where if it stays likely in both analyses, that might be something to prioritize fixing, even though it's a modified channel. And so let me walk you through some analyses where we have these sort of side-by-side results. You see results for similarly modified comparators versus all comparators. And I'm showing you just the natural channels to start. And there's really no difference. When you restrict your comparators to other natural channels, you get the exact same answer. You see some pretty different patterns, though, when you start looking at our different classes of modified channels. And again, I'm going to have you guys focus on the hard bottom channels at the bottom, where you see, first we said that almost all of them were were likely having a temperature or habitat problem. Temperature suddenly drops by half. So about half of them, we still see evidence for a temperature problem, but the rest of them, it's either indeterminate or unlikely. So this analysis of existing data, this is only one step that the SMC is doing to try to get at ways to fix modified channels. We're also trying to collect some new data to give us some new insights. And this is a part of the project that's actually gone through a lot of evolution since it started. We initially tried to look at conditions in restored modified channels, but we actually had a lot of trouble finding them. Most of the restoration activity, first of all, it's not very well consolidated. There's not like one database to find every restoration in the region. So we don't know where they're all occurring. Those that we could find mostly were not occurring in modified systems. These were often in natural channels, particularly in mitigation banks. So those weren't really going to tell us how restoration could work to fix these systems. Then we shifted to look at impacts of channel maintenance to see if there were certain types of patterns or maintenance activities that made a difference to what kind of scores we observed. But again, we had a hard time trying to find sites where we know there's a lot of maintenance going on, but it's not always documented in a way that helps us know when and where we can sample, just because it's just not recorded, with that kind of, projects in mind. So we're actually now moving on to a third phase of the study where we're focusing on high scoring modified channels. Our approach is that we're going to try to find, the highest scoring sites in each, county or watershed in different areas. These don't necessarily have to be, reference-like scores, but they're relatively high compared to other similarly modified channels. So we're talking about, CSCI scores typically ranging from 0.5 to 0.8, but not much higher. We're pairing them with lower scoring, similarly modified sites. And we're going to go to these sites and try to measure a wide range of stressors beyond the normal range. Given sort of the past evidence, we're really interested in temperature. So we're going to put out loggers, which will give us continuous measurements. This will provide us with a lot more insight than simple like spot measurements of temperature. You can really see the whole flux and we can tell, are these low scoring sites better? We're also going to look at pesticides and sediments, at least in soft bottom channels where you expect to find fine grained sediments. And when we have that data, we'll make these comparisons between, high and low scoring sites. And we assume we're going to find, different levels of stress that our high scoring sites have, maybe less temperature stress than we find at our low scoring sites. And this is what we're expecting to see. So next steps, again, we're going to be, taking another look at our rapid screening causal assessment, try to expand it. Like I said, we're not looking at all the different stress types that occur. You'll hear more from Katie about flow alteration. We're going to try to develop what we're calling a protective assessment framework to figure out, what's going on at high scoring channels, like what are the factors that contribute to high scores. And we do want to continue looking at restorations, even though we don't have a lot of sites. So if you've got some restored modified channels in mind that should be studied, please talk to me. For that, I'll take, how much time for questions? Just
6 minutes.
Okay, great. So plenty of time for questions. System that we've come up with for classifying as hard bottom and soft bottom. We did try to develop that on a statewide scale. So we do think it should apply to most regions of the state. We're particularly interested in trying to make sure it worked for urban as well as agricultural areas. In terms of the causal assessment approach, I would say it will work in any part of California, but we haven't really tested it well outside of Southern California. We actually have a new study by the Perennial Stream Assessment that Annie's been leading to sort of apply rapid screening causal assessment to streams in the Sierras and the North Coast. And so we've got sort of a first case test study that, hopefully we'll be talking about that next year.
Thank you. I just want to throw the pitch out there that the San Gabriel River Regional Monitoring Program, I think we'd be a great outlet for you to pilot some of this stuff. So there's funding.
Always glad to hear that. And a lot of these ideas actually were piloted in the San Gabriel. A lot of the causal assessment we first developed there. More questions or? Any? And nothing online? All right, well then let's get on to our next speaker. This is Dr. Katie Irving, my colleague here at SCWRP. And Katie will be talking about how we're looking at flow alteration in modified channels.
Thank you. Cool. Yeah, so I'm talking about flow ecology in modified channels. It is part of the same project, just a different flavor, I guess. And my name's Katie Irving, and I work here at SCWRP, for those that don't know me. Just to give you a bit of an outline of what I'll be talking about is I'm going to start off with an introduction to flow ecology, so you know what I'm talking about. And then I'm going to talk about flow alteration in modified channels. I'll talk a bit about the approach, some findings, and let you know what we have planned next. So what is flow ecology? It's basically a model that relates flow alteration to stream biota. And this is an example. This is taken from real data. So I'll show you later how we actually use that curve. But basically you have some kind of measurement of biointegrity on the y-axis. In this case, we have CSCI, can be a probability or anything else really. And then on the x-axis, it's a measure of flow alteration. And that means we define that as a change in flow or flow metric from baseline to current. So it's like the delta, it's the difference. So where you see 0 and the green dashed line, that's basically the expected CSCI score we would have under unaltered hydrology. So you can see that it's quite high. I don't know if I have a pointer, but you can see at the top of the curve, it's quite high. It's like 0.9 or something. And that's what we would expect. But we can use it in a way that we can get kind of flow ranges. So if we take the standard 0.79 for CSCI threshold and see where that hits the x-axis, these ranges here, we would need to stay within those to reach that standard CSCI threshold. So overall, the flow ecology analysis gives us a few different things. So first of all, it gives us an understanding of the influence of flow alteration on biology or biological condition. We can get flow ranges associated with good biology. And we can also understand what kind of seasonal flow components are important for the biology. And that third point comes from the type of metrics that we use. And we always use the functional flow metrics. These are a kind of the foundation of the California Environmental Flows Framework. And they're basically metrics that quantify the flow components, which you can see in kind of the makeshift hydrograph there. So we have wet season base flow, so winter base flow, summer base flow, dry season base flow, spring recession flow, peak magnitude flow, things like that. And every single one of those flow components has individual flow metrics that describe like the magnitude, timing, duration, frequency, and that kind of thing. So with regards to modified channels, our flow ecology models at the moment tend to be focused on trying to get back the reference conditions. And that's using that, for example, that 0.79 CSCI threshold. And that's probably not going to be appropriate for modified channels because it's just not going to reach there. So we don't want to be like fighting a losing battle. So it's basically that we need to build newer or updated flow ecology models, or at least make them adaptable for modified channels. And basically, this is how we do that. So as I mentioned, the most modified channels aren't going to reach that standard threshold. So we're going to modify the thresholds. And these are based on channel modification type, which Rafi kind of went through earlier. So that's the different types of the hard bottom, the soft bottom with 2 hardened sides or no hardened sides, or reference or natural channels. And we have a best observed score for each of these different categories. This is what actually Rafi presented on last year, I believe. I'm sure you all remember that very well. But just for an example, so the hard bottom channel at the bottom on the table is 0.67. So it does give kind of a bit more leeway. So that's what we're going to use for this analysis. And so, yes, this is what it looks like. So you recognize this curve from earlier. I told you it was real. But this, so we've just changed it to be more specific. So it's CSCI score. And this is for dry season base flow, which is measured in CFS. So this curve basically gives limits for both the depletion side, so the negative below 0, and the augmentation for each flow metric. So for example, this is how we get the flow limits. So for example, again, we do the 0.79 threshold. That basically means these limits here, we need to be above -9 and below 4 CFS in dry season base flow to get that threshold. And just for a kind of contrast, we can also look at the hard bottom, which was the 0.67. And the flow limits for the flow ranges we need to be between there is -16 and below 11 CFS in hard bottom channels to get that. Okay, so we applied those thresholds limits, the flow limits to all of the channels that we have data for. And I'm going to show you some results over the next couple of slides, but I just wanted to show you how we kind of categorized everything to make it easier to look at. So there's basically 4 different options based on whether it passes the biology and it passes the flow. So basically, as an example, if the biology is okay and the flow is within the limits, then we don't have to worry about those channels in this context. Also, if it's just one of them, the biology or the flow, we're not kind of concerned with those because it's generally not a flow problem, or at least not enough to be damaging the biology. But if we have channels where the biology is not okay and it's not within the flow limits, that's where there's potentially a flow problem. So this is the category that we're interested in. And we think that these channels here are really going to benefit for some flow management because that's going to improve or help to improve the condition within those channels. So the next couple of slides basically focus on this red box here, all the channels within that category. Okay, so start off with dry season base flow. So that's summer base flow. And this is the % of channels within that red box. So if you look at the hard bottom channel, which is the red column, that's nearly 75% of those channels potentially have a flow problem, enough that it's causing problems with the biology. So we really think that managing summer base flow is really going to help modified channels. Same with the soft bottom channels too, and even some of the natural channels. So just as a contrast, just to look at the wet season base flow, the % of channels within this category, within wet season base flow, was much lower. So we don't think managing wet season is actually going to help much in modified streams. Okay, so in terms of what's next, there are 2 main areas that we want to kind of focus attention. First of all is, well, as with any modeling project, there are some data limitations. We want to make sure that we have enough sites within each of the modified channel classes and they have a range of conditions. So we want to make sure that curve is as representative as possible. So it covers all bases. And the second area is, it's a little bit more complex. Because the flow alteration, the way that we use it is it actually ignores the interaction between the altered flow and the channel modification itself. And to do that, we need hydraulic models. And the reason that we need to consider it is, so the flow alteration metrics that we use are really good at like a regional scale. We've used them very successfully to kind of find out problem areas and prioritize. But they're basically based on the alteration that within the watershed rather than in the reach itself. So including hydraulics such as depth and velocity is really going to get at the local scale factors. So understand kind of how that's interacting. So for example, flow is going to move very much faster over concrete. It's going to be shallower than in like a natural substrate. And that's going to be what's kind of really going to be affecting biological condition. So that's definitely where we want to aim. So we're going to get a reach scale difference rather than just a watershed difference. So if you want to hear more about that, please come to me at lunchtime. And that's everything from me. Thank you very much for listening.
Thanks, Katie. That was really good. So what do we do about competing regulations? Where one of the things you mentioned on there was about improving dry season base flow, especially on that negative side, right? And competing that or contrasting that with some of the regulations which are saying we need to decrease dry weather flows or even eliminate dry weather flows into some of these urbanized channels. And how do you kind of balance those two objectives?
Yeah, that's a good question. I mean, I think it just depends on what trade-offs you want to. Oh, sorry. Yeah, it just depends on what else is going on, I think, in the channel. And yeah, we just got to find some trade-offs. It may be that we just have to say, maybe change the substrate. Maybe we need to do something else. You know, it may not be that we just directly have to turn off the tap, you know. We just got to kind of figure out what we can use causal assessment that will help that as well.
Thank you. Good talk, both of you. I have a question on if there's groundwater dependent ecosystems at all with the flow. And this might be ignorance from NorCal, but like the soft bottoms may have more of an interaction with groundwater than a hard bottom. And how's that impacting flow and also the summertime base flow? And if that's at all a concern for you guys. It is for us. We're always getting hammered with GDEs. And so I'm just wondering if there's anything available for this that can translate. Thank you.
Yeah, good question. So yeah, you're right that groundwater is going to influence, especially in the soft bottom reaches. We don't include it in the flow ecology analysis as yet, but it's definitely something that we want to work on kind of moving forward. But yeah, but obviously in the hard bottom, it's not going to penetrate through. But yes, definitely something we're looking into. Hey.
Thank you. Yeah, really good presentation. It was cool that you were able to find like optimal periods where you can like restore the habitats. What do you think you could what could be done to restore these habitats to like have higher CSCI scores?
Like in addition to the flow, or yeah, how would you improve the flow? It depends on what else is going on in the channel, because it may be that there's a temperature problem. There may be that it's you know, the substrate problem or the habitat directly. So it really depends on what is going on. Yeah. Oh, okay.
Okay, we have a question online from Jonathan Martin. It says, with the whiplash precipitation hydrology regime, the majority of our coastal watersheds experience, are the natural conditions of our streams better represented as a bimodal distribution around too dry, too wet, and that maybe less than 30% of the time we have suitable hydro conditions to support more diverse, robust benthic communities?
I'm not sure if I got all of that, to be
I can connect you with him online and you guys can have a better discussion. Yeah. He says, I'm sorry. Yes, please. So I'll, yeah. No problem. And let's see, I got a anonymous question. How does effluent dominated watersheds factor into both the CSCI and flow analysis? Many SoCal waterways, example, San Gabriel River, have elevated oxygen and temperatures due to high percent effluent flows in addition to being line.
Yeah, I'll take a quick answer in terms of the causal assessment and research that I presented and add to it. So it doesn't directly get incorporated because we're using the CSCI as an assessment tool, and it can tell us whether or not these effluent dominated channels have reference-like biology or don't have reference-like biology. And I think it really works well for that purpose. And the causal assessment can then tell you whether the conductivity or temperatures are likely contributing to poor biology. I think it's a totally separate and valid question about what are the appropriate goals for those types of systems, particularly as we have competing uses with, ecological benefits as well as, water reclamation. So I think that's something that a lot of folks in this room are very concerned about. So I'll pass.
That's
Apologies. You have anything to add?
yeah, I mean, the thing with flow ecology, we are already looking at flow, and we have to make that very, very clear. We know that temperature and everything else is going to be a factor too. We want to include those in future models, but we are just looking at flow and looking at those very separately. Hey.
I can just talk loud.
Okay.
No, use the mic, please. Okay, I'll use the mic. Okay. Thank you. My name is Mike. I'm from the Santa Ana Water Board, and thank you for the presentation. I just wanted to clarify on your last slide about next steps or something. Was I understanding you correctly that you're going to look more at velocity as an indicator of biointegrity instead of flow, because flow depends on the channel cross-sectional area as well. Is that kind of what you were saying? In a sense, it's like a what's it called? Velocity ecology instead of flow ecology.
Yeah, right. So yeah, I would call it a kind of hydraulic ecology model. Yeah, so it would look directly at the depth and the velocity. The main reason being that describes the channel within the cross section. The flow metrics that we use are going to be very dependent on what happens upstream and going down. So that's why it's kind of based on the watershed scale rather than the channel scale.